Okay, sorry again. Thank you, Brianne. Thank you, everyone, for your patience. Yep, we kind of get it going. Yeah, so today I have the pleasure to share with you some of our research in the topics of modulating glycosylation as a potential approach in the development of treatments for neurodegenerative disease. And this research of mine really originates from a fundamental basic research in the laboratory where I'm working with Professor David Catlos that eventually leads to some preclinical findings that has implications in a clinical stage right now. So, but yeah, I need to make a disclosure of conflict of interest. Some of the data that I'm going to show you today involve some of the early OJ inhibitor technology that was licensed by SFU to a spin-off company known as Electos Therapeutics, where my supervisor, Professor Catlos, served as a founder, shareholder, and he's currently a consulting chief scientific officer at the Scientific Advisory Board. Okay, since today we have an audience with a really diverse background, I thought it would be good to kind of remind everyone how we as molecular scientists look at the definitions of life. And it's really based on four major classes of biomolecules as shown here. We have these nucleic acid that forms our DNA where our genetic information is encoded. And based on our genes, we have these expression of these proteins that are really the players within the cells carrying out all of these functions and all of these activities driving cellular processes. And then on these sides, on the other sides here, we have these cheesecake-looking like structure that is lipid, which is best known for its function as a barrier segregating between compartments. And last but not least, we have carbohydrate, best known for being a source of energy, but it can also bind to protein and lipids and modulate their properties and functions. Now, today we're going to focus mainly on proteins and carbohydrates. And as mentioned, proteins are basically direct products of our genome, right? We start from the nucleus where our DNA gets opened up, serve as a template for to create the generation of this temporary copy of your gene that is known as mRNA. And it can escape the nucleus and enter the cytoplasm, be recruited at this organelles called ribosomes, where the ribosomes will read this mRNA while recruiting the corresponding tRNA carrying this amino acid to be disembarked at this site here. This keeps cycling until we finish reading the entire mRNA, and you have a whole chain of amino acid. Now, at the same time, this amino acid chain will also undergo folding due to molecular interactions, eventually forming into these mature three-dimensional globs, where it's at this mature state of proteins that can now be directed to the corresponding locations for carrying out its functions. Now, that's not the entire story to dictate the functions of a mature protein. When proteins are mature, they are going to be susceptible to a number of modifications known as post-translational modifications. And this diagram here really shows you major classes of these modifications. And for most of them, it's really about adding a chemical unit to certain locations of the protein to modulate their functions and properties. Now, those of you who have backgrounds in biology would be very familiar with the term phosphorylations. This is by adding the phosphate group to certain site of the proteins, and is mostly catalyzed by a specific class of enzyme called kinase that put this phosphate group onto proteins, and is most commonly seen in this kind of signaling pathway where proteins interact with one each other to carry out downstream functions. Now, it turns out that having too much of this phosphorylation actually contributes to the underlying pathologies that is commonly seen as a hallmark for Alzheimer's and Parkinson's disease. In the context of Alzheimer's disease, we have this neuronal protein called tau that can undergo excessive, or we call it hyperphosphorylations, that will eventually lead to the formation of tau aggregate known as neurofibrillary tangles. And in the context of Parkinson's disease, the presynaptic protein called alpha-synuclein can also undergo hyperphosphorylations and form aggregate that is known as Lewy bodies. And they follow very similar kind of aggregation cascades as summarized in this diagram here. You start off with a healthy form of these proteins, usually at the soluble state. But under cellular environmental stress or even traumatic cause, it undergoes hyperphosphorylations, and now it becomes sticky. And it can recruit its own kind to form the initial aggregate known as oligomers and start entering this pathological cascade. And this serves to facilitate further aggregations to elongate these aggregate into something called protofibrils. And when you get to a certain density of these protofibrils, they start clumping up, tangling with each other, and eventually leads to the formation of mature aggregates. And at this stage, this aggregate is insoluble and very difficult to clear and induce long-term damage to the brain. So the general theme of the current kind of development for therapeutics really try to slow down this aggregation cascade, hopefully at an earlier stage to slow down the progression of these neurodegenerative diseases. Now, it turns out that there's one post-translational modification that acts on similar site as phosphorylation that has been shown to antagonize this process. And it is this O-glucosinylation, or we can call it O-glucoside modifications, which involve putting this sugar unit onto the protein. Now, this sugar unit is synthesized by taking up a portion of glucose that's taken up into the brains. So it is recognized as a nutrient-sensitive product. And what stood out for this particular sugar unit compared to other sugar molecules is that while other sugar attachments can usually be found on the surface of cellular membrane, most of these O-glucosinide sugar modification can actually be found within the cell, in the cytoplasm and the nucleus as well. So it really draws the attentions of the research community at the time. Now, if you see this and you feel overwhelmed, like, am I going to go through all of this? No. But I do want to show this to illustrate that there are many targets that could be susceptible to O-glucosinide modifications. And this is only a fraction of thousands of targets that can be modified by this sugar. But despite having all of these targets, there's really only two enzymes that regulate this process. You have the enzyme OGT that puts the sugar onto the protein. And on the other hand, you also have these OGA enzymes that can cut the sugar off, restoring the protein to its unmodified state. So it is the fact that you have a very simple regulatory mechanism that acts on so many targets that makes this particular sugar modifications simple, but also one of the most poorly understood in the field. So some of the early findings on the topic of O-glucosinide modification actually suggest that it might have an implications on preserving brain health. And there was a paper published in 2004 that looks at the O-glucosinide modifications in patients with Alzheimer's disease. And they're able to see that these patients typically have a lower level of O-glucosinide modified proteins in their brain compared to individuals without the disease. And what's striking was that in this particular paper, they reported seeing no sugar modifications of the tau protein that engage in these tau aggregations in the diseased brain at all. Now, this coincidentally aligns with some of the early clinical findings using PET imaging technology when they were measuring the uptake of these glucose analog attached with radioactive probe. They're able to see that for individuals who are carriers of genetic mutations who will develop the inherited form of Alzheimer's disease, there's a general reduction in this glucose uptake compared to healthy individuals without the mutations, even during the time when they're still cognitively intact. So all of these findings kind of suggest that perhaps there's a neuroprotective effects of O-glucosylation, and perhaps if we're able to develop some chemical tools to block the activities of OGA from preventing it from removing this O-glucosinide sugar modification from proteins, will increase the level of O-glucosinide proteins in the brain, in turn reducing the aggregations of proteins as the potential disease modification strategies. So the laboratory at the time works on kind of synthesizing some of these inhibitors, and after a couple of generations, will land on these particular molecules known as thymic G. It has a couple of properties. First of all, it's a small molecule inhibitor, meaning it can reach to many places of the body, which is typically difficult to reach. And it was designed to be very potent and specific to binding and inhibiting OGA enzyme, one analogy that I've seen my supervisor giving in the presentation was like, if you put a couple milligrams of Thymosin G in an Olympic-sized swimming pool, and then you have OGA enzyme in it, it is going to inhibit the enzyme. So it kind of illustrates how potent it can be. But most importantly, it is the blood-brain barrier penetrant, meaning it can cross the blood-brain barrier, which is a lining of cells that really barricade molecules from the bloodstream from entering the brain, and it's very highly selective. So this molecule is readily available to enter the brain through the bloodstream. And because of all of these properties, the laboratory at the time was able to conduct some of the earliest preclinical studies to test these strategies. So they were able to dissolve these Thymosin G compounds in drinking water and treating into these mouse lines that is known to accumulate tau aggregations in the brain and develop Alzheimer's disease. So after 8 to 9 months of treatment, they collect the brain tissues and analyze the amount of the sugar modifications in the brain tissue using biochemical technique. So this figures in the center here is showing you a Western blot experiment where we typically use to measure these sugar modifications. You can see mice that was treated with Thymosin G has more of these sugar modifications compared to mice without the treatment. Now, when we analyze the tau aggregates using histological technique, actually it might be a little bit hard to see here. So you can see from the cohort that has the sugar modification, which is shown in these green panels, these green here, the tau aggregate shown in red here has a strong reduction compared to the cohort without receiving the treatment, which is reflected in this bar chart on the top. So this presents some of the earliest findings on OGA inhibitor antagonizing the pathologies associated with Alzheimer's disease, and similar findings have been replicated and discovered by other research groups using the same inhibitors in other mouse models of Alzheimer's disease, or even using other OGA inhibitor. Now, when I first joined the laboratory, we were interested to look at how O-glucanate modifications might provide other neuroprotective benefits through other cellular pathways, given the fact that O-glucanate acts on so many targets, involving in so many pathways. And one pathway that really drew our attention at the time was endocytosis, or basically taking up materials from outside of cells within the cells. And this is because it was found at the time that these protein aggregates can actually be taken up by healthy cells and then starting a pathological cascade in these cells, eventually killing those neurons. And it's also been reported in this pretty old study that there's a reduction in this sugar modification for a specific protein that is known to regulate this kind of molecular trafficking within the cells in patients with Alzheimer's disease. So we hypothesized that perhaps O-glucanate modification can influence the uptake of these toxic protein aggregates and then slowing them from entering into cells. To investigate these mechanisms, we use cell culture study. So on a culture dish, we culture these cells of neuronal lineage, and then we treat them with this Thymosin G inhibitor at single concentrations and incubating for a multiple period of time to investigate a potential time-dependent effect. When we use the Western blot technique that you have seen earlier to measure these sugar modifications, we're able to see a time-dependent increase of this modification indicated in this bar chart below. Now, when we repeat the experiments, but this time at the end of the treatment, we deliver these synthetic alpha-synuclein aggregate, which is shown in this red punta here, we see an opposite direction. We see a time-dependent reduction in the uptake of these alpha-synuclein aggregates in these cells. We also investigate potential dose-dependent effects by incubating these cells with different concentrations of this inhibitor for the same amount, for the same period of time. And when we use Western blot to measure the sugar modification, we're also able to see a dose-dependent increase. Now, when we put in the synthetic synuclein aggregates at the end of treatment and we incubate them, we're also able to see for cells that receive the highest dosage tested in these experiments, they also show a dose-dependent reduction in this uptake. So our findings demonstrate time-dependent and dose-dependent properties of these OGA inhibitor in reducing the uptake of these toxic protein aggregates that really motivate us to pursue a preclinical studies by administrating this thymogen compound into a mouse models of Parkinson's disease. This M-Thy1-HSNCA mouse line is known to overexpress human alpha-synuclein, which will in turn accumulate these alpha-synuclein aggregate at regions of the brain that are susceptible to synuclein pathologies in the context of Parkinson's disease. So we treat these mice with 2 dosage of the thymogen. We have the higher dose at 500 mg, and then we have the lower dose at 200 mg, and then we have vehicle without any thymogen treatment. So we treat them for 10 months, and at the end, we collect their brain tissues and do biochemical analysis. As control, we use the wild type C57 black 6 mice, and then treat them with the high dosage of thymogen and with vehicle. When we collect their brain tissues and analyze the sugar modifications using Western blot, we're able to see for both mouse strain, there's a significant significant increase in the level of sugar modification as shown in these quantifications here. And the cohort that received higher dosage seems to have an uptrend toward having more of these sugar modifications compared to cohort receiving the lower dose, though the data didn't reach statistical significance. But what's strikingly was that when we analyze these alpha-synuclein aggregate, which is shown in this green panta here at the substantia nigra, which is the vulnerable regions for synuclein pathologies in Parkinson's disease, we're able to see that both of these, the dosages for both the low and high dosages of thymogen leads to a significant reduction in the number of these aggregates at this region. And especially in cohort receiving the high doses of thymogen, there is a significant reduction in the size of this aggregate compared to vehicle-treated cohort, indicating a potential dose-dependent effect. So collectively, our cell culture and preclinical data really points to OGA inhibitor being a viable disease-modifying approach, where similar findings have actually been reported by other research groups using other mouse models of Parkinson's disease and using other OGA inhibitor. And together, we're able to show that OGA inhibitor could potentially be pursued in clinical trials as a potential treatment for Parkinson's disease. Now, our journey on OGA inhibitor doesn't just stop there. We are currently exploring potential beneficial effects of OGA inhibitor in kind of antagonizing the pathological drivers that may lead to protein aggregations. And one of these drivers is repeated low energy traumatic brain injury or repeated mild traumatic brain injury. Because it is found in individuals who have a career or have frequent exposures to repeated head trauma that they are more likely to develop dementia, including Alzheimer's disease and something known as a chronic traumatic encephalopathy, or abbreviated CTE, as they retire. And where the tau aggregations happens to be the major pathological features of these neurological disorders. So we want to see how OGA inhibitor may be able to kind of slow the progressions of these pathologies using preclinical mouse model. So we collaborate with Professor Cheryl Willington's research group over at the University of British Columbia, who developed a really sophisticated TBI model called the Chimera TBI. So our next speaker is going to talk a lot more about Chimera, so do stay in line. But in general, we are able to deliver concussive surgery-free type of TBI into these mouse lines that also accumulate tau aggregates in their brain. So when we deliver these TBI, repeated TBI at a mild energy settings within 2 weeks at a very young age, and then we age these mice for 6 months before we collect their brain tissues for histological analysis of tau pathologies, we're able to see at some of these cortical regions that are susceptible to neurological injuries and Alzheimer's disease that there's a chronic increase in the level of these total tau proteins, and as well as a chronic increase of these tau aggregates at these particular regions. validating that our TBI models can exacerbate tau pathologies in this particular mouse strain. So based on these findings, we are motivated to pursue the ongoing dosing studies where we perform the same TBI paradigm using the exact same mouse strain, but incorporating this thymoG treatment starting at the second TBI of the procedure. We're going to treat these mice for 6 months, and then at the end, we're going to collect their brain tissue and blood samples, and we're going to look at tau pathologies, as well as other biomarkers associated with neurological injuries and neuroinflammation. So I would like to take the last bit of this presentation to kind of follow up with you, like how far it has gone to regarding OJ inhibitor being in the clinic. So since the first introduction of OJ inhibitor, there have been several groups in the industry and some in academia as well, developing their own OJ inhibitor, and some of which actually reached clinical trials as a treatment for tau pathology related to Alzheimer's disease and other neurological disorders known as the progressive supranuclear palsy. As shown here, they have reached several stages of clinical trial at the moment. And on the right here, I'm showing you some of the data released by the pharmaceutical company Eli Lilly, reporting their most recently wrapped up phase 2 clinical trials on their OJ inhibitor, sapiranostat, where they treat patients who are diagnosed with early symptomatic Alzheimer's disease with this OJ inhibitor for around 1.5 years. And by using volumetric MRI, they're able to see at the end of the treatment, there is a slowing of this loss of brain mass, as shown in this top part here, compared to patients receiving only placebo treatment. Now, they also measure the levels of these plasma phospho-tau 217 biomarkers, and as well as GFAP, which are biomarkers that have currently been developed clinically to be a fairly sensitive biomarkers indicating tau pathologies in the brain and neuroinflammations. They were able to see that participants receiving the treatment within the span of treatment actually demonstrate lower level of these biomarkers, indicating reducing tau pathologies and neuroinflammations. So there's still more work to be done to kind of optimize this OJ inhibitor as for it to be completely ready to be used in the clinic. But I think this kind of shows you more evidence that O-gluconate modification or OJ inhibitor has the potential to provide additional neuroprotective effect other than just countering tau pathology. And together with other preclinical studies that we have done and other groups have found, really motivate further investigation and effort and resources into developing OJ inhibitor as a potential treatment for a wider range of neurodegenerative disease. So I would like to conclude by, hopefully I will be able to kind of convince you with this short little talk that O-gluconate modification is a very common post-translational modifications. Despite having so many targets in the cells, it's only being regulated by 2 enzymes, which makes it a very interesting and still quite poorly understood modification so far. And in our hands, we're able to show using OJ inhibitors that it is able to significantly increase the level of brain O-gluconate while reducing the level of toxic protein aggregates in mouse models of Alzheimer's and Parkinson's disease. And the several neuroprotective benefits that have been shown by our group, by other groups, and also in clinical studies really warrants further development for OJ inhibitor to be a disease-modifying treatment target for various neurodegenerative diseases. So yeah, lastly, I would like to thank everyone who have contributed a key role behind the data that I was able to show you here today. I would like to thank our collaborators at UBC for the ongoing studies. And we'd also like to thank the funding agencies for their continued support. Yep, this is for Callo Group. We are a laboratory, a hybrid lab with very talented chemists and biologists working together to crack down these really difficult topics. And last but not least, thank you, INN, for supporting me in my trip to an international conference to share OGA findings to a broader community. Thank you so much.
Thank you, Jeffrey, for that wonderful presentation. And it is now my pleasure to introduce our second Trainee Award winner, Victoria Carriquiriborde, and I have the pleasure of being her PhD supervisor. Victoria used the INN Travel Award to attend the Society for Neuroscience meeting in Washington, D.C. last year. And she is a PhD candidate in the cognitive and neurosciences area in psychology. So welcome, Victoria. We look forward to your presentation.
Thank you so much, Dr. Kent, for the presentation, our introduction. Today, I'm going to be presenting on some parts of my PhD project on the effects of repetitive concussions on sleep in a mouse model of Alzheimer's disease. And thank you, the Institute for Neuroscience and Neurotechnology, for letting us come and give this talk today. It's a privilege. Before I start, I want to respectfully acknowledge the ancestral and unceded territories of the Coast Salish people, including the Tsleil-Waututh, the Coquitlam, Musqueam, and Squamish nations in which the SFU Burnaby campus resides. I am privileged to be able to do my research here and spend most of my time in these lands surrounded by forests, mountains, and nature. So most of you know that I am a rugby player. I have been playing rugby for 6 or 7 years now, and I've seen my fair share of concussions. But this is not the only contact sport that include concussions. For the Canadian fans here, hockey is a major contact sport that also has high risk of concussions. Skiing also experience concussions, and even soccer players experience concussions. So this is a topic that is very dear to my heart, and I think it's very important to study because it clearly affects a lot of people, not only in sports, but also in the day-to-day activities. All right, so concussions are a type of traumatic brain injury. And a traumatic brain injury refers to a brain injury that is caused by an outside force. It can be divided into closed and open traumatic brain injuries, where open traumatic brain injuries are less common and more severe, and closed traumatic brain injuries are a lot more common. It's something that you would see with falls or motor vehicle accidents, sport injuries, or blast injuries, and they range in the severity. Within the closed injuries, or TBIs, we can further classify them according to the type of acceleration that the head and the brain kind of is subjected to. This can be translational, rotational, or angular accelerations. So the translational type, the head will move in a straight line, and this usually results in a more focal injury. While the rotational acceleration, the head will rotate in its axis, resulting in a diffuse shear strain, and this usually causes that diffuse axonal injury that is so common in TBIs. Lastly, the angular type is a combination of both the rotational and linear accelerations, where the head moves in an angular manner. And the angular is the most common one and accounts for a bulk of the TBI cases. It is important to understand the TBI biomechanics because depending on the models of TBI that we use in animal research, we might be kind of recreating some types or different types of concussions in humans too. For the most part, I will be talking about closed head injuries in my presentation. So traumatic brain injuries can also be divided according to their severity between mild, what we usually call a concussion, moderate, and severe. 80% of our TBI cases are mild TBI cases, and it is estimated that 200,000 concussions are sustained annually in Canada. This criteria to assess severity of TBI is most commonly based off consciousness or alterations in consciousness, post-traumatic amnesia, and the Glasgow Coma Scale score. So sometimes when there is a moderate or severe TBI, they might also include some structural imaging too, to see if there's any structural damage in the brain, but this is less common for mild cases. And note that even when you have a mild TBI, this can cause a loss of consciousness of up to 30 minutes. So there's quite a range of severity even within the mild TBIs. I'm going to be mostly focusing on this severity range for my project. So after you get a TBI, this can cause a cascade of cellular and molecular events in the brain that can include an increase in blood barrier permeability, increase in neuroinflammation, impair axonal transport, and neurotransmitter imbalances and synaptic imbalances. Most of these effects are present during the acute phase, but some changes can be long-term. And additionally, repetitive mild TBIs can increase the risk of developing chronic traumatic encephalopathy, as Jeff mentioned, which is a neurodegenerative disease that is linked to repeated trauma to the head and can lead to dementia. Repetitive mild TBIs have also been associated with the most common type of dementia, which is Alzheimer's disease. Now, Alzheimer's disease accounts for 60 to 80% of all of the dementia cases. It is a progressive neurodegenerative disorder that primarily affects older adults. And the prevalence of AD is rapidly increasing due to the aging population and our longer life expectancies. The pathology of AD is characterized by the accumulation of amyloid beta plaques and neurofibrillary tangles in the hippocampus. And as we know, the hippocampus is kind of a main hub for memory processes. AD can be classified also into mild, moderate, and severe. And these severities are based on symptoms such as the degree of memory loss, behavioral symptom changes, and motor impairments. And there's also some loss of independence associated with the disease. AD can be classified also by onset. So early onset, also called familial AD, appears early in life, around mid-30s, mid-60s. And it's mostly related to genetic mutations, whereas on the other hand, late onset AD typically affects people age 65 and older. And etiology is the complex interplay of aging, genetics, metabolic, and environmental factors. Currently, the diagnosis of AD uses various blood and cerebrospinal fluid tests, neurological examinations, and brain imaging. CSF levels of A-beta and p-tau are the two most common used in biomarkers for the diagnosis of AD. However, therapy for AD is majorly directed at the alleviation of symptoms. We don't have a cure yet. And the development of disease-modifying treatments that can slow or halt the disease progression is the major research goal still. Now, before I kind of continue with my presentation, I wanted to give a little bit of a background on electroencephalogram and kind of how it works, because I know that not everyone is familiar with this method. So the electroencephalogram, or EEG, measures and records electrical brain activity in the brain. Furthermore, quantitative EEG is a type of EEG analysis that involves processing, transforming, and analyzing the EEG signal using complex mathematical algorithms. And QEEG has allowed new ways of extracting signal information. For example, analysis of specific frequency bands and signal complexity, analysis of connectivity and network. And the analysis of specific frequency bands is very common in EEG, as you can see. I think this is, there we go, here. We have, these bands are associated with distinct brain states and sometimes during specific functions. So for example, the gamma and beta bands, which are here, are considered high frequency bands, and they're mostly mostly related to attention and kind of things that we're doing when we are awake. The alpha band is prevalent when we are relaxed or in a sleepy state, and usually ranges from 8 to 13 hertz, whereas the theta and the delta bands, which are these ones here, are mostly related to sleep stages and can range between 4 and 8 hertz and 0.5 to 4 hertz, respectively. Now, there we go. EEG analysis has helped researchers learn about brain function during sleep. We know that in humans, we have the sleep cycles that are around 90 minutes long, and that during the sleep cycles, we go through different stages of non-rapid eye movement, non-REM, and rapid eye movement, REM. Non-rapid eye movement is further divided into N1, N2, and N3, and these stages are kind of deeper in sleep. The stages are associated with different quantitative EEG characteristics. For example, the theta frequencies are dominant during N1. This is mostly for humans. N2 is defined by the presence of sleep spindles and K complexes, and also sharp wave ripples can occur during non-REM. And these particular features of non-REM have been associated with sleep-dependent memory consolidation. In N3 of non-REM, this is characterized by the delta waves, the slow waves, and slow waves have been linked to learning and memory functions, immunity, and clearance of metabolic waste in the brain. Therefore, sometimes this is known as restorative sleep. Lastly, REM is characterized by brain activity that is pretty similar to wake, and this is when we have our most lucid dreams, and is accompanied by muscle paralysis, which stops us from enacting those dreams. REM is also linked to memory function processing of emotional memories and has been associated with dementia, with less REM associated with more risk of dementia. Usually N2 and N3 stages, which are the deeper stages of sleep, have longer durations during the beginning of the night and get shorter and shorter by the end of the night. And on the other hand, REM is the opposite, where it's shorter during the beginning of the night and longer as the night continues. There are normal changes in sleep as we age. For example, shorter durations, more frequent awakenings, and decrease in deep sleep. However, there are some adverse changes in slow wave sleep and REM that have been associated with Alzheimer's disease. The link between Alzheimer's disease and sleep is not completely clear, but there are some studies that suggest that sleep could play a role in the development of or worsening of AD pathology. As mentioned before, sleep is implicated in clearing of brain metabolic waste. Multiple studies have shown, both in mice and in humans, have shown that sleep deprivation can cause an increase in the accumulation of A-beta in the cerebrospinal fluid, and how these levels can decrease once sleep is allowed. They also have shown how A-beta accumulates throughout the day. So the longer that you're awake, the more A-beta that you have in your system. There are multiple ways in which sleep can interact directly or indirectly with the neurodegenerative processes, but more research is needed to understand the role of sleep in neurodegeneration and specifically on Alzheimer's disease. Moreover, one of the most commonly reported symptoms after TBIs, especially mild TBIs, are sleep complaints. Hypersomnia is very likely to be present in patients within the first week after the injury, whereas insomnia symptoms are more prevalent after the first week. And insomnia symptoms can last up to 2 to 3 years after the TBI. So that's quite a long time. It is important to study the effects of mild TBIs on sleep because sleep impairments can have consequences on the recovery trajectory of repetitive mild TBIs and lead to more issues in the long term. And as previously mentioned, sleep is also related to Alzheimer's disease. So that is why at the aim of my study was to investigate how repetitive mild traumatic brain injuries, or RMTBIs, impact sleep in a mouse model of Alzheimer's disease at different stages in the pathological progression. So to do this, we used the closed head impact model of engineered rotational acceleration, or chimera. And you can see the machine here, where the mouse is laying belly up, and then the head is aligned with a hole that has a piston that will come up and impact the head, and the head is freely allowed to move. So therefore, the advantage of using this chimera model is that it combines the impact factor with this rotational and linear acceleration. So it's kind of like an angular acceleration replication of what a TBI would be if you were hit with something and your head was allowed to move, or you're in a car accident. Then the mice received a stereotactic surgery where we implanted 2 screw electrodes to record electrical activity in the brain and an electromyographic wire, or EMG, to record muscle movement. Note that the electrodes were coming from inside the skull, so this is sometimes called electrocortical recordings, compared with the use of the scalp EEG in humans. So it's a little bit more invasive. Brain activity was recorded for 72 hours, and then we used the last 24 hours for analysis. The last 24 hours were broken down into 10-second epochs, and each epoch was classified as one of the 3 vigilance states: non-REM, REM, or wake. And here we have a little bit of an example of what these stages look in animals or mice. So in mice, wake EEG is characterized by higher frequency activity in the frequency spectrum, lower amplitude in the raw EEG, and higher muscle activity. Whereas non-REM is characterized by higher power in the lower frequencies in the delta band, higher amplitude, and lower muscle activity. Finally, REM is characterized by a higher power at the theta frequency range. That's 4 to 7 hertz, lower amplitudes, and almost no muscle movement. The overall project had 3 cohorts of mice that received the mild repetitive TBIs at 6 months old. The cohort 1 was then recorded right after TBI and once every week for 4 weeks. Cohort 2 was recorded 1 month after TBI, and cohort 3 was recorded 9 months after TBI. For this presentation, I'm going to be showing data for this first cohort of mice, so when we recorded right after TBI. But before I do that, I want to explain why we chose the 6 months old time point to deliver the TBIs. So for our study, we used the APP-NLF knock-in mouse model of Alzheimer's disease. And from previous studies, we know that in these animals, at 6 months, plaques are only starting to develop, and the plaque load is very low. In these images, you can see that even at hopefully you can see, yeah, that even at 9 months, plaques are very sporadically distributed across the brain. And it's only around 12 to 15 months old that plaque accumulation in the brain really grows exponentially. So we chose 6 months because we wanted to see if repetitive concussions would have an effect in the development of this plaque formation. So here's kind of a schematic of what our animals went through. First, they had their TBIs. Males received 3 TBIs, females received 2. And this is because of previous findings of mortality issues and morbidity issues with the females. And then the day that they received the last TBI, they also received the stereotactic surgery. And right after that stereotactic surgery, we connected them to the recording chambers and recorded them for 72 hours. After that, every week for 4 more weeks, we recorded 48 hours of EEG data. All right, up to the results. So here I'm showing the percentage of time spent in each vigilance state for each EEG recording session. And there's a little bit of like a cheat sheet here explaining what the sessions mean. So session 1 would be 48 to 72 hours post-TBI. Session 2 is 7 days post-TBI. Session 3 is 2 weeks after TBI. Session 4, 3 weeks, and session 5, 4 weeks after TBI. So those are the sessions here in the x-axis. And then in the y-axis, we have the percentage of time spent in that particular vigilance state. On the top, I also put some examples of how our data looks like for wake in the yellow, for non-REM in the blue, and for REM in the red. And it's also color-coded here. Hopefully you can see the wake. So what we found is that there was a significant interaction between time and treatment, both for wake and for non-REM. But this was only, the statistical significance was only a time effect. So basically, the mice spent less time in wake in the first session compared to all of the other sessions, and more time in non-REM in the first session compared to all the other sessions. There were no significant changes in REM. So what this is showing is kind of like that time effect. And one of the things I suspect this effect is from is the fact that both sham and TBI went over this surgery and they were under anesthesia for 40 minutes or so. So I'm thinking maybe there's some effects of this surgery that can last up to 48 or 72 hours after the procedure. And so they're affecting both cohorts or groups the same way, and maybe perhaps masking those effects from TBI. But then within the other sessions, it kind of goes back to baseline. We also were interested in sleep fragmentation. We know from AD patients and as well as TBI patients that they experience this fragmented sleep during the night, so more awakenings during the night, or they also kind of complain about having to nap more or feeling like they're tired. So we're thinking, okay, maybe that's what's happening with these mice. They are just coming in and out of the states more often. So we look at the number of transitions between the vigilance states. So first, for the non-REM to REM transition and REM to wake transition, so this third column, what we found is, again, on the first session, they seem to be transitioning less between the stages compared to all of the other sessions. However, when we look at the transitions between non-REM and wake and non-REM, they are transitioning more between these states in the first session compared to the rest of the sessions. So there seems to be some sort of sleep fragmentation after that first, within the 48 to 72 hours after the procedure, although this is for both groups, sham and TBI. So they these mice might be having this broken sleep during the day. And we also were interested in these quantitative EEG measures of power spectra. So this is the normalized power spectra for the frontal electrode during wake. On our y-axis, we have our frequencies, which is 0 to 30. Sorry, it is so small. You gotta believe me there. And then on the y-axis, we have power on microvolts per second square. And this is normalized for all the mice. So what we see here during wake, the TBI animals seem to have lower brain activity within the theta range, so around 4 to 7 hertz, compared to the sham group. And this is true for session 1, but then it kind of goes away for the rest of the sessions. There are some trends on it, but the groups that have a lot of variability, like within the group, there's a lot of variability. So this effect seems to be present only in this first session. And we know that during wake, mice have more activity or power within the theta alpha frequencies. So lower levels of activity in the TBI group could mean that while awake, the TBI group is functioning at a lower capacity than the sham group. We also looked at this power spectrographs for non-REM, and we found that there was no differences between the TBI and the sham group, and no differences between the sessions. And we also looked at REM, oh no, at the power spectra, the normalized power spectra at REM state. And again, there was no differences between the TBI and the sham group. They all seem to have equal levels of power across all the frequencies. And this was the same for all the sessions. So in conclusion, after repetitive mild TBIs, mice had similar amounts of sleep compared to the sham mice. Mice spent more time in non-REM and less time in REM or wake during the 48 to 72 hours after mild TBIs compared to the following sessions. Sleep was more fragmented within the first 72 hours compared to the other sessions, and there was a decrease in brain activity in the repetitive mild TBI group compared to the sham group during wake within the theta band. And this concludes my presentation. I want to acknowledge and thank my awesome supervisor, Dr. Brianne, also my collaborator, Jeffrey, who had an awesome presentation before, and Dr. Sean Talk, who's no longer in the lab, but helped me tremendously with this project. Also, our collaborators at the University of British Columbia, all of the arrays that have helped me out with all the data that we have collected, our sponsors, the Western Brain Institute, and finally our SFU animal facility for keeping our mice healthy and safe for the most part. Thank you.
Well, please join me in thanking Victoria and Jeffrey for their wonderful presentations.
Thank you, everyone.
Thank you both. And don't forget to apply for the INN Travel Awards, trainees.